Student Lára, Stefansson, examiner and guests, welcome to this MSc examination. I will now in Icelandic explain to the audience how the examination will proceed, and then I will turn back to English. Fyrir hönd rannsóknarnefndar, námsnefndar, læknadeildar býð ykkur velkomin. Hér mun nú fara fram meistarapróf þar sem að Lára Stefansson fjallar verkefni sitt, samspil MITF og frumurhingsins í sortuæxli, sem á ensku heitir The Relationship Between MITF and Cell Cycle in Melanoma. Umsjónarkennari og leiðbeinandi Láru Eiríkur Steingrímsson og aðrir í meistaranefnd eru Margrét Helga Ögmundsdóttir og Valerí Fokk. Prófari í dag prófessor Guðmundur Hrafn Guðmundsson. Sjálf heiti ég Jóna Friðsdóttir og fyrir hönd rannsóknarnefndar læknadeildar mun stýra þessu prófi. Prófið fer þannig fram að Lára Anna hefur 30 mínútur til að kynna verkefni sitt og svo tekur Guðmundur Hrafn við. Og hann hefur alltaf klukkutíma en 30-40 mínútur bara alveg hæfilegt, en ég mun stoppa hann eftir klukkustund. Síðan gefinn kostur á stuttum spurningum frá ykkur áhrendum og eftir það förum við, prófari og leiðbeinandi og ræðum prófið og komum loks aftur og tilkynnum niðurstöðuna. So now I will turn back to English. Dear Lára, you have 30 minutes to present your project. Afterwards, Guðmundur has up to an hour to examine, and then the examiner, your supervisor and myself will go outside and decide on the outcome of this examination and come back here and present the results. So the floor is yours now.
Hi, I'm very excited to present my master's project to you guys. My master's project is called The Relationship Between MITF and Cell Cycle in Melanoma. So first, melanocytes. They are cells that originate from the neural crest and then migrate to the skin. Shown in this diagram here is the epidermis, which is the outermost layer of the skin. As you can see, closer to the surface of the skin, most of the cells are keratinocytes, but as we move deeper into the skin, there are more cell types. And shown here are melanocytes at basal membrane of the epidermis. These are melanin-producing cells. Melanin is the pigment that gives skin their color. Melanin is produced in organelles called melanosomes, and then they are transported to the keratinocytes where they have a protective effect from DNA damage. So when melanocytes become malignant, it results in melanoma. Melanoma, well, the incidence has risen 3 to 8% per year, with most cases being people of European heritage. The melanoma is associated with sun exposure, or more specifically ultraviolet or UV exposure. So wear your sunscreen. Melanoma is one of the most highly mutated, heterogeneous, and lethal forms of cancer. In a study done by the Cancer Genome Atlas, over 300 melanoma samples were taken, and their mutations were characterized. And in over half of these samples, there was a mutation in a kinase called BRAF. More specifically, the mutation was BRAF V600E, in which the valine is replaced with a glutamate at position 600. And this causes a hyperactive form of BRAF, which then causes downstream effects. And one of the proteins that is affected by this mutant is MITF. So before I go into the role of MITF in melanoma, what is MITF? MITF is microphthalmia-associated transcription factor. Its structure is shown here as a basic helix, loop helix leucine zipper. It can either homodimerize with itself or heterodimerize with another member of the MIT family, such as TFEB, TFE3, and TFEC. MITF is known as the master regulator of melanocytes, as it is important for regulation of genes important for pigmentation, cell survival, apoptosis, autophagy, and cell cycle. So MITF does play a pretty crucial role in melanoma. For the melanomas with the overactive BRAF V600E mutation, MITF is known as a lineage survival oncogene, meaning these cells need MITF in order to survive and proliferate. And there even is a mutation that is associated with melanoma called MITF E318K, in which the glutamate is replaced with a lysine at position 318. This causes a decrease of simulation of MITF, and this change in a post-translational modification increases the transcriptional activity of MITF, and people with this mutation of MITF have a 5 times higher risk of developing melanoma compared to the general population. So I'm going to switch a little bit and talk about cell cycle. So cell cycle is the process by which cells grow and divide. It's normally broken down into 2 phases. There's interphase and mitosis. And then interphase can also be broken down into G1, S, and G2. It's a diagram shown here. During G1, the cells are mainly growing and getting ready for DNA synthesis, which takes place during the synthesis or S phase. Following that is G2, in which the cells continue to grow, and then they prepare for mitosis, which is the process by which the cells actually divide and become 2 cells. There's also a stage that we call G0, which is shown here, in which the cell is not really growing or preparing to divide, it is merely just existing. There are many genes and proteins that regulate the progression through cell cycle, but kind of the most well-known and mostly studied are cyclins and cyclin-dependent kinases, or CDKs. They are shown here in this diagram. They are synthesized and degraded at different points of the cell cycle, and when they are active, they form a complex like this, which then help regulate the progression through the cell cycle. Also shown in this diagram are the cyclin and CDK inhibitors, P16, P21, and P27, which are also used to regulate the cell cycle. Now, in this thesis, I will be defining the cell cycle stages based on how much DNA is in the cell at that time. So during G1, there's only 1 set of DNA. During S phase, there's anywhere between 1 and 2 sets of DNA. And then both in G2 and mitosis, there are 2 sets of DNA. Because of this, I will be grouping G2 and M together. I'll be calling it G2/M. So to bring these 2 topics together, MITF is known to regulate cell cycle. One of the biggest ones that we know of is that MITF directly binds to and activates CDK2, which was actually shown in this diagram, both here and here. And it is known for this G1 to S transition. MITF also regulates P16, P21, P27, which were the cyclin and CDK inhibitors shown in the diagram before. Shown in this diagram here is a very simplified signaling pathway to MITF. It shows Wnt signaling, CREB, and MAPK all going down to regulate MITF. They do this either transcriptionally or post-translationally. Most known is that MAPK phosphorylates MITF at position, serine at position 73, which then regulates MITF through its transcriptional activity. And then MITF, in turn, after being regulated by these pathways, then go to regulate pathways important for differentiation, apoptosis, and cell cycle progression. The purpose of my thesis is not just to figure out how MITF is impacting cell cycle progression, but how cell cycle can then come back around and impact MITF. So there is something to be studied. There was data done by some members of the lab. One member of the lab was able to generate a MITF knockout cell line using CRISPR-Cas9 technology. Then another member of the lab was able to generate a list of differentially expressed genes shown here. And what I am most interested in are the differentially expressed genes that are cell cycle related. Most, namely being the CDK5 and ABL enzyme substrate 1, or KBLS1, cyclin-dependent kinase, or CDK2, leucine zipper tumor suppressor 1, LCTS1, and cell division cycle 16, or CDC16. So I had three main aims for my project. The first is to explore MITF throughout the cell cycle. The second is to explore effects of MITF on cell cycle regulating genes, mainly the ones that I have just mentioned. And then I took two of these cell cycle regulating genes, and then I checked to see what they do to MITF. So for my first aim, I wanted to see what happens to MITF throughout the cell cycle. What happens to its phosphorylation status? What happens to its expression level? But in order to do that, I needed a method to synchronize the cells. So what I did was something called a double thymidine block. It is a very common method for synchronizing cells. And so thymidine is a DNA synthesis inhibitor. So I basically did 2 overnight treatments with a release in between. And then I released the cells from the treatment. And then in theory, because they're unable to go through DNA synthesis, they would all be in G1 when I released them, and then they would all continue together throughout the cell cycle, and I can see what MITF is doing. So was this successful? Shown on the left here is the cell cycle profile determined by FACS for each time point. Shown here is unsynchronized. I don't know how well you guys can see it, but there's kind of an even spread of G1, S, and G2/M. And then at 0 hour time point, the majority of cells are in G1, except for here. And then after 4 hours, a lot of them are in S right here. Then at 8, the majority of cells are in G2, and at 12 hours, they're mainly back to being in G1. So this first shows that the synchronization worked. And then secondly, that every time point more or less corresponds to a cell cycle stage. Both at time point 0 and 12, the cells are mainly in G1. At 4 hours, they're mainly in S phase, and then at 8 hours, they're mainly in G2/M. And shown here on the right is a Western blot. I stained for MITF, cyclin B1, and beta tubulin, and these are the protein levels at each time point. Beta tubulin is a loading control. Cyclin B1 is used as a cell cycle stage marker, and it stains for cells in G2. And this pretty much just confirms, since the most intense band is here at 8 hours, this confirms what we see here, that most of the cells are in G2/M. So what is happening with MITF? You can see that the bands are not terribly intense at 0 hours. They get a little more intense and are the most intense after 8 hours of treatment, and then it kind of goes back down at 12 hours. And another thing to note is the double band pattern of MITF. It is well documented that the upper band here is the version of MITF that is phosphorylated at serine 73, and then the bottom band is the version that is not. So what I did to determine phosphorylation status of MITF was using the upper to lower band ratio. So I quantified this Western blot, shown here. So this shows the upper to lower band ratio of MITF throughout the time course. As we see, there is a significant decrease in phosphorylation, or of the ratio, at 0 hours, which might indicate that there was a dephosphorylation of MITF at G1. However, if this was the case, we would see it again at 12 hours, but we actually see the opposite. Compared to the 0 hour time point, the 12 hour time point is significantly larger. So this would show that this effect here, it has probably less to do with G1 and more to do with the thymidine treatment. Shown here is a quantification of both the upper and lower bands of MITF, and these are compared to the unsynchronized control. And the pattern that I saw was consistent within the 3 repeats, and at 8 hours there was significantly more MITF protein compared to the unsynchronized control, as well as the 0 hour time point. So the next question was, is this increase in MITF protein due to an increase in MITF transcription? So what I did is I checked RNA expression of MITF throughout the cell cycle, and as you can see, there's really no big difference from time point to time point, meaning that the increase seen here is probably not due to increase in transcription. So then the next question was, if I do something to MITF, how does that impact cell cycle? So what I did was I used a cell line generated in the lab that uses a DOX-inducible expression of microRNAs that knock down MITF in this cell line 501 mel, which is the same cell line that I had used for the previous experiment. So this slide shows the synchronization and the two cell lines. Here's the control and the knockdown. So I both induce the cells to knock down MITF and then synchronize them to see what happens to the cell cycle progression when I knock down MITF. Shown here, the bands are very distinct in the control, and then they go away quite a bit. You can still see the double band pattern, but to me this looks like an efficient knockdown, and I can use this to determine the differences between the two. So what happened? Here I'm showing you the unsynchronized cell cycle profile determined by flow. I am using the unsynchronized profile to show essentially what are the general trends of the cells. If they spend more time in G1, it will show up in the unsynchronized control. If they spend more time elsewhere, it also shows stuff. There is actually a method in which the percentage of S can be used to estimate proliferation of the cells. So if the cells are more in S phase, it's more likely that that batch of cells is proliferating faster. So what is the difference between the two? There's really only one. You see more G1 here. It's a difference of like about 6%. And this increase here is taken both from S and G2/M. So the question was, when I knock down MITF, is there a G1 arrest? Or is there a difficulty for G1 to S transition? So there's a buildup of G1. So in order to do that, I looked at the profiles from each time point. Shown here, the cell cycle profiles after each time point between the control and the knockdown cell line. And I know you can't see this well, but you see it as well as I do. There's no difference between the two. If there was a difficulty for the cells to transition from G1 to S, we would see that at 4 hours, when most of the cells are supposed to be in S phase. There is quite a lot of S phase here, and it's pretty much the same here. So this would indicate that there's no difficulty transitioning from G1 to S. Instead, there's just generally a G1 arrest upon MITF knockdown. And then on the flip side, what happens when I overexpress MITF? So I used a different cell line. Well, it's still 501 melanoma cells, but now there is a DOX-inducible expression of GFP-tagged MITF. And shown here is the Western blot, which is stained for GFP. So note that this is the added MITF. There is still the endogenous amount, but this is the amount of overexpressed MITF in these cell lines. And this showed that the overexpression works, and I can probably use it to determine what happens when the cells have so much MITF. So again, with the unsynchronized profiles, the biggest difference here is probably the S phase, and then there's more S phase here, and also less G2. So this could mean that the cells are just generally proliferating faster. It could also mean that they're spending less time in G2/M. So I looked at the cell cycle progression for these two, and both at time point 0 and 4, they're pretty much the same, mostly in S, mostly G1 here. But at 8 hours post-treatment, the S phase is this much, and then for the overexpression cell line, it's not much. There is a reduction of S phase at 8 hours after the treatment, and then at 12 hours you see that the overexpression cells have pretty much gone back to what they were at the 0 hour time point, while the empty vector control has not quite made it yet. So to summarize these experiments, there was an increase in MITF protein during G2/M. There is a slight trend for more G1 upon MITF knockdown, and this is likely due to general G1 arrest of the cells. And there is a slight trend for more S phase and less G2/M phase on MITF overexpression, and this might be because of faster proliferation and also just general faster progression through G2/M. So on to my next aim, to explore the effects of MITF on cell cycle regulating genes. I used the same DOX-inducible cell line that I had used previously. And something to note about 501 melanoma cells, they have a generally high endogenous amount of MITF. So knocking down, there's usually a lot of MITF, and then they have to adjust to low. From the Western blot before, we know that there is an efficient knockdown of MITF protein. So what I did was I treated the cells with doxycycline for 48 hours, and then I checked for actin, MITF, and then these 4 genes that I had mentioned previously. And then I also checked MCM2, which is also a cell cycle regulated gene. I normalized both to actin and to the control cell line. What did I see? There was a significant decrease in the amount of MITF, which was nice to see. I already knew that the knockdown of protein was good, but the knockdown of RNA was also fine. There was a significant decrease in cables 1, and then also a decrease in CDK2, which we expected because it is known that MITF binds and activates CDK2. This was not significant, but the p-value was 0.07, so with one more repeat, this could potentially become significant. And to look at the opposite, I used a different cell line. I did not use 501 mels, instead I used 8375Ps. And this cell line has a DOX-inducible flag-tagged MITF. And something to note about 8375P, they generally have a low endogenous amount of MITF. I had previously used these cell lines, so I knew that there was an efficient overexpression of the protein, but I did not have a Western blot to prove this. What I did was a doxycycline treatment for 24 hours, and then I checked for the same genes previously, and then I normalized to actin and the MD vector control. And what I found was not much. There was an increase in MITF, the lowest amount being 2-fold to highest being 5. So that seems like almost enough to determine the effects of overexpression of MITF on these genes. There was a general trend for all of these to increase in expression, but again, nothing was significant. To summarize, cables 1 decreases significantly upon MITF knockdown on 501 melanoma cells, and CDK2 also decreases. And there's no significant difference among the genes checked when I overexpressed MITF in 8375P cells. There was a general trend for the expression increase. One thing to note, it is very difficult to compare these results because the role that MITF plays in these 2 cell lines are likely very different, since 501 mels generally have a high amount of MITF and 8375Ps generally have a lower amount of MITF. More repeats would be necessary to further determine the effects. So on to my last aim. I took 2 of the genes that I checked and then wanted to see their effects on MITF. So to give another little introduction, CDK2, cyclin-dependent kinase 2, is a serine threonine kinase. It is known for its role in G1S transition, and we know that it is directly regulated by MITF. So what I was interested in is, so MITF regulates CDK2, does CDK2 then come back around and regulate MITF in a feedback loop? And the other gene that I chose was leucine zipper tumor suppressor 1. And this is not the structure of LZTS1, it is the structure of the complex that it interacts with, which is the CDK1 cyclin B1 complex. And this complex plays a role in mitosis. And in cells lacking LZTS1, they have faster progression through mitosis. And LZTS1 is decreased in uveal melanoma. So because there is an association with melanoma, and this protein plays a role in G2/M, and there seems to be something with MITF and G2/M, as there's an increase of MITF protein G2/M. I was interested if these two proteins somehow interact, and outside of our lab there hasn't really been much research to show the connection, so this was super interesting to me. So what I did was I knocked down either CDK2 or LZTS1 in 501 melanoma cells, and shown here is the Western blot, and visually you can see that upon knockdown of CDK2 or LZTS1, there is a decrease in MITF protein. Now, this was consistent among the 3 repeats, as shown by the quantification here of the Western blot. There was significant decrease in MITF protein both after knocking down CDK2 or LZTS1. Now, what's interesting is when I knocked down these 2 genes and then I checked RNA expression of MITF, there was only a significant decrease when I knocked down CDK2. However, when I knocked down LZTS1, there is not a significant decrease of RNA expression of MITF. So, to summarize, MITF RNA and protein levels decrease significantly upon CDK2 knockdown, and this supports the idea of a feedback loop in which MITF regulates CDK2, and then CDK2 then goes back around and somehow regulates transcription of MITF. And then only MITF protein levels decrease significantly upon LZTS1 knockdown, and this could indicate that LZTS1 has an impact either on the translational activity of MITF or the stability of MITF. So to summarize my entire talk and make my conclusions, G2/M is correlated with higher levels of MITF protein, and this is not due to an increase of transcription of MITF during G2/M. MITF knockdown causes a slight G1 arrest. MITF overexpression causes faster progression through G2/M. Cables 1 decreases upon MITF knockdown in 501 mel cells. CDK2 potentially regulates MITF transcription in a feedback loop, and LZTS1 potentially plays a role in translation and or stability of MITF. I'd like to thank Eirik Stefansson for giving me the opportunity to work on this project in his lab. I would also like to thank Margaret for always being positive and uplifting in my committee. I'd also like to thank Brooklyn for helping me with my flow data, and Valerie, Ronnie, and Josue for generating my cell lines. Everybody else at the ES lab, I couldn't have done it without each and every one of you, and thank you all for coming to this talk.
I was so interested to listen to this and forgot that I was counting this. But I would like to thank you, Lára, for a good lecture. And now I would like to ask the examiner, Gunnhildur Rapp, to come and ask her about the project.
Thank you. And thank you for a very